feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, hello, and welcome to the National University of Singapore Yong Lu Lin School of Medicine's Healthy Longevity webinar. Thanks for joining us again. It's going to be an exciting show. We have Professor David Gems tonight, who's not only known for his really elegant work in C. elegans, but he's one of the deep thinkers in the aging field. So hopefully we'll have a lot of things to discuss. I want to remind you to use the Q&A function to ask questions. There should be some interesting questions tonight, so pick out some good ones. Tonight, before we do that, we have one of our most notorious graduate students, Max, joining us from the Center for Healthy Longevity, and he'll be talking about his project on lipid clocks.
Thank you very much, and good evening, everybody. As mentioned, I'm Max. I'm a PhD student at the Kennedy Lab, and I'm working on the intersection of biological aging, lipidomics, and artificial intelligence. And today I'm going to tell you a little bit more about some of my work on lipid clocks. But maybe let's start out, what are lipids? Lipids are a heterogeneous class of hydrophobic and amphipathic molecules that in a way means they don't like water. And in your daily life, the most lipids that you come across are like fats and oils, which are just a subgroup of lipids. But you have them everywhere, like in your butter, in your ghee, in your bacon, in your fish, in your vegetable oil, in your nuts. When you eat something, also in the avocados. Other part of lipids that you might have heard of is when you go to a doctor and get your blood panel done and he tells you about cholesterol or like your hormones such as testosterone and estrogen. And in our body, lipids play like an important role. First and foremost, they're like for energy storage, right? That's what your fat is about. That's the weekly stuff. But they're very important for like keeping the structure of our cell membranes, but also act as signal molecules, signaling between tissues and different cells, telling the body what's happening inside. And for the frequent listeners of this webinar, you have heard like a lot of other people already talk about different kind of aging clocks, like epigenetic clocks, glycomic clocks, protamic clocks. And today I'm going to tell you about a lipid clock that we developed. And we developed this lipid clock on the blood serum of 800 patients, and it contains a lipidome of 148 lipids. What you see here on this slide is on the left panel, on the x-axis, you see the chronological age, and on the y-axis, you see the lipid age. That is the age predicted by the lipid clock. In green, you see like healthy people. And in red, you see people with pancreatic ductal adenocarcinoma, which is like the most common and also lethal pancreatic cancer, with like a 5-year survival rate of only 6%. And the first thing that strikes out is like that people that have cancer age faster. That's why their slope is higher. But on a population level, people with like pancreatic cancer have like a lipid age acceleration that is 8 years higher than for healthy people. And if we look at that more, we can see that lipid age acceleration is associated with like shorter lifespan in people with cancer. In total, people that have like a lipid age acceleration compared to people that have like a lipid age deceleration, people with lipid age acceleration have like a 73% higher chance of dying at any given time. If we look what the lipid clock is actually thinking, which lipids are important, we find that it mainly captures lipids that are already associated with like longevity, centenarians, or cancers in literature. So there's very neat that this lipid clock can map back to biology, and we can learn from that of what is really happening in the organism. Moreover, if we do like lipid pathway analysis and compare young people to old people that are healthy, and also compare people with like decelerated and accelerated lipid age, we find that in both cases, sphingolipid metabolism is like really dysregulated. And that tells us probably something about the mortality dynamics, and that there's a similarity between cancer and normal aging mortality dynamics. So in conclusion, lipids play an important role in aging and cancer. Lipid aging clocks can provide insights into the aging process, and lipids can be potential targets or supplements to modulate the aging process. And with that, I hand it back. Thank you very much.
Thanks, Max. So tonight we have David Gems. He's a professor of biogerontology at the Institute for Healthy Aging at University College London, where he studies the biology of aging. His work is on the nematode C. elegans and has contributed to the development of new programmatic theories of aging. Tonight, his topic is bad spandrels, biologic constraint as a cause of aging. David, it's good to see you. It's been a while.
Yeah, it's been too long. And so, hello, everyone. Good morning from London, and good evening to folks in Singapore. So yes, I work on worms, on C. elegans, and also on theory. And I'm going to talk today about the theory. So this is going to be pretty much a data-free talk. So I thought I would start by asking a general question. I think my impression of the field over the last decade is that there's been a kind of a drift away from the idea that aging is actually something that you can really understand in general terms. One sort of figure that expresses this, I think, quite nicely is this from this very excellent review from Brian, which includes this idea of the pillars of aging. And if you look at this figure and think of it in terms of the possibility of understanding from first principles what the causes of aging are, this figure kind of tells you just to despair and give up, that there's sort of no possibility of really understanding aging. And I come across this quite a lot. It's not everybody, but I would argue very strongly that aging has to be understandable, and we can't really progress really effectively in the biology of aging without some sort of general basic understanding of aging. And as Brian said, yes, there's a kind of a whole framework of ideas that's been emerging over the last decade, including the programmatic theory. And that's what I'm going to talk about today. I'm going to talk about part of that theory, as sort of a newer part. And this was something that I put together very much during the lockdown in the pandemic. And I should say this is work very much done in a collaboration with a very smart postdoc in my lab, Karina Khan. So I'm going to really talk about what's in this rather complex theory paper. So at the heart of the paper is a question about the phenomenon of antagonistic pleiotropy, what exactly it is and how it causes aging. And this is it's kind of an unsolved and very central part of aging theory. So some of you will be familiar with this concept if you're from the aging field, but let's go over it again. It's was originally put forward by George Williams, American evolutionary biologist. And it's a theory about the evolution of aging, why aging evolves, why it exists. The argument is that many genes are pleiotropic in nature, which is to say they have effects on multiple different traits. And sometimes you'll get a gene or a new allele that has both beneficial and harmful effects on the organism. And so those are antagonistic effects. And Williams argued that because the force of natural selection declines with age, if you have these alleles with good and bad effects, and the bad effects only appear later, then natural selection may actually favor them because of the greater importance of beneficial effects during youth. So to put it in a kind of a simpler terms, what evolution is really doing is playing nasty buy now, pay later games, right? Where you get early benefits and later costs, which come in the form of diseases of aging. And in terms of understanding primary causes of aging, an important prediction of the antagonistic pleiotropy, the evolutionary theory in general, is that a major cause of aging, a primary cause of aging, is actually the wild-type genome. It's a wild-type gene action. So this theory has been around for many years, more than half a century, and it's a very well proven theory empirically. But it has remained largely an evolutionary theory. So presumably you have these genes with antagonistic pleiotropy that cause aging, and they're acting on some kind of proximate or kind of downstream actual biological mechanisms. And those are the mechanisms that actually give rise to aging and give rise to the diseases of aging, which are to a large extent the same thing. And this is the thing which has remained really unclear. So really what I'm going to talk about is ideas about how the action of antagonistic pleiotropy genes cause aging and how aging evolves. So let me start by saying something about antagonistic pleiotropy a bit more broadly, what it is. So it's actually a very, it's a phenomenon that exists in many forms, not just the form that Williams envisaged, which is shown here in this cartoon. So in the cartoon, you see this bar shows kind of a life history of an organism with senescence on the right. And you can see a gene having two effects. It's having a beneficial effect shown by a round symbol, and then a detrimental effect later shown by a sort of bomb symbol. But there are many different kinds of antagonistic pleiotropy, all of which involve essentially a situation where you have a gene which is in one context having a good effect, another context having a bad effect. So, for example, you can have a gene which has a good effect that's dependent on the genotype. So, for example, maybe if you have one copy of the gene, that's beneficial, and you have two copies, that's harmful, as in a sickle cell anemia, for example. Or you may have a gene which is beneficial in one sex, but harmful in another. And then there are many examples of genes where the effects are environment dependent, right? So maybe, for example, you have a gene where you have low levels of food, it's beneficial, high levels of food, it's harmful, gives you diabetes. Or maybe it's beneficial in the presence of pathogens, but not the absence, or in the presence of predators, but not in the absence. So many different kinds of antagonistic pleiotropy. So as part of this this essay, I went out to get a list of examples of antagonistic pleiotropy to work on. Kind of curious about how many I could find, and I got to more than 100 before I gave up. So this is a very ubiquitous phenomenon, antagonistic pleiotropy. Many genes have been found that have this property. But I should emphasize that antagonistic pleiotropy is only detectable in these cases because there's variation in the gene, right? If you've got two variants of the gene, you can identify the antagonistic pleiotropy. But one would expect many genes where you only have a single variant, in which case you can't actually detect the antagonistic pleiotropy. So the examples that we find are almost certainly the tip of the iceberg. So this is highly ubiquitous. Right, so now we come to the kind of core of the theory. And there's no equations or anything in this. This is all kind of a deductive theory. So why is antagonistic pleiotropy such a common characteristic of genes? It's very, very common. And here is our central hypothesis. So this looks a little bit like Brian's pillars diagram, but it's something quite different. So what this shows is a group of traits in an organism, and the fact that those traits are interconnected with one another. And this is because biological systems are very highly integrated. What that means, so the size of the circle is an indication of the sort of magnitude of the fitness. Okay, so here we have a mutation and selection, and you can see that two of these traits have gotten bigger. They've become improved. But then because of the connections to all of many other biological characteristics, you also get a reduction in fitness in other traits. So what this is arguing is that there's a constraint on the optimization of multiple traits because they're interconnected. So this is biological constraint. So let me sort of go a little further to sort of explain this idea of biological constraint with a kind of a sort of semi-real example. It's a hypothetical example, but based on real biology. And this is ATP synthesis, right? So for thermodynamic reasons, if you increase ATP synthesis rate, you reduce the yield, and vice versa. If you lower the rate, you can increase the yield. So imagine if you have a mutation, so that it's constrained. You can't maximize both. The properties of ATP synthesis are constrained. So let's say you have a new allele, and it's increasing ATP synthesis rate, and that's favored by natural selection. So that allele will exhibit antagonistic pleiotropy because it's going to reduce ATP yield, right? So what this this sort of shows how antagonistic pleiotropy can arise from the presence of biological constraint. It also tells us something, I think, quite fundamental about antagonistic pleiotropy, which is that it doesn't really arise from the properties of genes per se. It's not like something to do with the molecular function of the gene so much, a sort of property of the gene. It's more a property of the constrained systems that those genes are affecting, right? So what this then, if constraints are so important for understanding antagonistic pleiotropy, then it's very important to understand the nature of constraint. And here we drew a lot on a really excellent review, having looked for sort of theory papers about the nature of biological constraint. The best one that we found was from Luis Acerenza, he's based in Uruguay, and he was trained in Edinburgh in Scotland. And he put together a sort of typology of constraint, which we then adapted into this scheme of different kinds of constraint. So I'm not going to go into this in detail. I'm just going to say that the interconnection constraint is one type of constraint, and I'm largely going to talk about that. But there's another kind of constraint called impossibility constraint, which I'll mention briefly. So, and again, this is drawing on Acerenza and kind of expanding his arguments. As he noted, biological constraint is something that exists at pretty much all levels of biological organization, right from the level of the gene, for example, with linkage disequilibrium on chromosomes, where you have genes that are genetically linked to one another, and that can lead to something what John Maynard Smith called genetic hitchhiking. So you have a selection for one, selection for a particular allele, and genes that are nearby will be co-selected with it. So that's an example of constraint. And then you have all these other kinds of constraint, which I'm not going to go through. I think signaling constraint is probably a particularly important one in terms of where antagonistic pleiotropy comes from, right up to the multi-level constraints, where you've got constraints acting between effects on fitness at the individual level and sort of colony or deem, so that's population level. So there are many different sources of antagonistic pleiotropy from these many different kinds of constraint. So that in a way, the sort of the next question, the deeper question, is really, how does constraint give rise to antagonistic pleiotropy in aging? And this is something I'm only going to go into in a sort of fairly superficial way. It's quite a big topic. But to do that, I'm going to need to say a little bit about this programmatic theory that Brian alluded to there. So this is a theory that really has its origins in ideas from George Williams, and was developed, the sort of core of it was developed in the mid-noughties, pretty much simultaneously by Misha Blagascloni in the US and Pedro de Magalhães in the UK. And for those interested, I put out a sort of a overview of this theory last year in the Aging Research Reviews. So this is the basic gist of this idea, is that the main cause of aging is not, as had long been supposed, kind of random stochastic damage accumulation, but it's actually more the action of genes, the wild type genes, in later life. So this is, according to this view, wild type gene function is actively causing aging. It's not a passive sort of decay process like you get in machines. It's more something more programmatic. And to use Blagascloni's term, rather than it being a kind of loss of function, it's really too much function. It's actively caused. So he calls it, he uses the term hyperfunction. It's sometimes called the hyperfunction theory. And Pedro emphasizes more the importance of continuation of sort of developmental processes, including kind of adult developmental type of processes as well, not just ontogenetic processes in later life. As Misha, to again use Misha's terminology, what you have is biological programs that have evolved to promote fitness, but in later life, they continue on in a way which is futile and cause pathology. He calls them quasi-programs, which just simply means that they're genetically programmed, but they're not a program for aging, because the evolutionary theory argues that there is no program for aging. Aging is not adaptive. So he calls them quasi-programs. So here's an example, taking the things that I've spoken about, putting them together, the programmatic theory, hyperfunction, and the constraint theory, to try to make sense of antagonistic pleiotropy. So here's a real life example of antagonistic pleiotropy from this big table in the paper, and this involves a gene called AAT1, and this is a protein called alpha-1 antitrypsin. And this is alleles of this gene affect the likelihood of developing one of the most common causes of death in older people, which is COPD. So here we go. It's a bit complicated, this one, not too much. So AAT1, what it is, it's an inhibitor of an enzyme called elastase. Now, elastase, it does various things, but one of the things it does, it's secreted by neutrophils in order to enable them to bore through tissue to sites of infection, to tackle infection. And in the lung, this elastase has a kind of like a collateral injury effect. It actually injures the tissue that it bores through. And that contributes in later life to the development of COPD. So what we have with neutrophil elastase is a constraint on optimization of its function. Because on the one hand, if you have more neutrophil elastase, that's going to improve resistance to infection. It's going to help the neutrophils to go tearing through tissue to the site of infection and prevent death from infection. But on the other hand, in later life, the long-term consequences of the elastase is to cause deterioration of lung tissue and to contribute to COPD. Okay, so now we get to the actual real antagonistic pleiotropy example. So we have variation at the AAT1 gene. So there's a rare allele of AAT1 called the Z allele, and that has reduced levels of AAT1. So what that's going to do is to, one would expect that to increase the elastase activity and to improve resistance to infection, but increase increase susceptibility to COPD. So this allele also has another effect, which is that it reduces the frequency of ischemic heart attack, especially in early life. It's not clear why that happens. It's sort of been speculated that it may be that the elastase is helping to increase elasticity somehow in blood vessels and reducing hypertension, so reducing heart attack. So we've got AAT1 exhibiting antagonistic pleiotropy. So the common allele, the normal allele, as the Z allele shows us, is actually increasing risk of heart attack, right? Because this Z allele is reducing it. So that means that the common allele is hyperfunctional with respect to AAT1. But the Z allele, which protects against heart attack, is hypofunctional with respect to COPD. So hypofunction is a term introduced recently by Alex Matlakoff and Tracy Chapman from the University of East Anglia. It's a very useful term. So this shows you how you can understand antagonistic pleiotropy in terms of constraint and hyperfunction and hyperfunction. Very briefly, to say something about impossibility constraint. So this is something taken straight from Asarenza. To illustrate, so he uses this example. So if you take two molecules of ethanol and convert them into butanol, that's an exergonic reaction. So that would mean that if you have an enzyme, let's call it butanol synthase, that reaction would run very well, and one would expect that this would be a useful biosynthetic capacity for bacteria. But butanol synthase doesn't exist. So he argued that the reason why it doesn't exist is probably simply that it's not possible for it to evolve. The existing repertoire of active sites doesn't provide a route to its evolution. So sometimes traits don't evolve because it's simply not possible for them to evolve. So to use just one very the simplest example I've got of impossibility constraints affecting aging-related disease, let's look at gout. So gout largely affects older men. I don't think I need to explain what gout is. Well, it's uric acid crystal accumulation. You get this painful inflammation, especially in this joint in the big toe. So most mammals have an enzyme called uricase, which means that they don't accumulate uric acid. But the higher primate lineage has lost uricase. The gene has been lost. So even though there might be selective pressure to favor the presence of uricase, because we've lost it, there's no way of getting it back. So this is an example of hypofunction resulting from impossibility constraint leading to pathology. So that's a sort of a kind of a lesser cause of diseases of aging. But there are other examples of this. Okay, so now I'm going to come to the sort of the kind of final point, which relates to the title of this talk, which may have puzzled some of you. So I come to this via Venice, and this is the Plaza di San Marco, St. Mark's Square, and this is the Basilica of San Marco. And it was visiting this basilica that inspired Stephen Jay Gould, the evolutionary theorist and paleontologist, to write this very, very famous essay with Richard Lewontin called The Spandrels of San Marco. So here's what this is about. I really recommend this to anyone who hasn't read it. Fascinating essay. So he was looking up under the dome at the spandrels. So spandrels, it's an architectural term that describes the sort of triangular, basically a triangular space above where you have two arches together. Okay, and he was admiring the paintings of saints and other holy figures on the spandrels. And what he reasoned, he was thinking that the architect for the cathedral, when he designed the cathedral, he didn't specifically design it to have spandrels. The spandrels were simply a consequence of the fact that he put four arches together under the dome, right? And Gould used this as an analogy for how evolution sometimes works, which is that sometimes you get you'll get selection for some traits, and you'll get other traits that appear as a kind of unintended, if you like, consequence of that selection. And one example that Gould uses to illustrate that is the panda's thumb. So pandas, they have a sort of normal pentadactyl limb, but they've also evolved an extra digit from a bone near the wrist, which they use for holding onto bamboo. So that's evolved. But interestingly, if you look at the feet of pandas, you can see that the equivalent bone in the foot also has a kind of protrusion. So in other words, the selection for the evolution of this thumb has led kind of by accident, or rather in an unintended way, if you can put it that way, to a change in the foot. And this presumably is because of a developmental constraint. The program that makes the hand and the feet are closely related. So if one of them changes, the other one will also change. And there are quite a lot of examples of these spandrels in biology. A nice simple one is nipples on men, which is a sexual dimorphism constraint. They serve no function. They're simply a kind of a leftover from embryonic developmental changes in the embryo before you get separation of male and female traits. Another one, a slightly more controversial one, is this is a female hyena, and you see this thing hanging down there. This is called the pseudopenis, and it's been suggested that this may originally have evolved as a result of selection for aggression in hyenas that's led to a sort of a spandrel-like expansion of the clitoris of the hyena. So Williams, going back to Williams, he was familiar with the spandrel concept and was quite happy with it. And here's his definition: a structure arising as an incidental consequence of some evolutionary change. So we argue that one can expand the idea of a spandrel to root it in the idea in the concept of biological constraint. So broadly, spandrels are features that are non-selected consequences of biological constraint. Now, here we can make an interesting connection. So Williams, in his original 1957 essay, he actually once mentioned an idea about how perhaps antagonistic pleiotropy might work in terms of proximate mechanisms. So he imagined a gene involved in calcium deposition. So this is like, I guess, a gain of function allele. And so this would then improve bone growth. It will accelerate bone growth by increasing calcium uptake into bone. You can imagine a baby zebra, the bone's growing faster and it can escape from hyenas or cheetahs or something like this. But then the same gene in later life is causing calcium deposition into arteries and contributes to cardiovascular disease. And in fact, this fits in completely the definition of a spandrel, according to our expanded definition. And Williams, surprisingly, didn't spot that connection. It's also an example of a quasi program, right? It's a program for calcium deposition that carries on in a futile fashion, contributes to pathology. Very different to the old disposable soma theory, which is another sort of ultimate proximate theory. Right. So to sort of conclude, the bad consequences of antagonistic pleiotropy, one can understand as being bad spandrels. So, going back to the classic essay from Gould and Lewontin, it's rather like the spandrels of San Marco, but replacing the saints and other holy figures with sort of skeletons and demons. So what this is arguing is that to at least to a considerable extent, it would predict that that diseases of aging one can understand as essentially bad spandrels. Okay, so to conclude, antagonistic pleiotropy arises from interconnection constraint in particular. Impossibility constraint also contributes to the evolution of aging, and the unselected consequences of antagonistic pleiotropy, including much of what contributes to late life disease, one can understand as spandrels. I haven't really, I should thank all these people for all the worm work mostly, but I haven't really talked about that. But I should very much thank Karina Kern, who worked with me on the theoretical aspects, including some of this constraints story. Just to mention another output from the lockdown. This is a book which I just finished about a month ago, which will hopefully come out in 2024. This whole programmatic theory and the kind of wider framework of ideas is really fascinating and exciting. So I sort of put it all into this book, which is written in a kind of very accessible and hopefully sort of a educated layperson type of level of reading. So it should be hopefully useful for people who are interested in this. And that's it, really. So I've covered some of what's in this article, but certainly not all of it. I hope it might have whetted people's interest in it. It's currently still only a preprint. And I leave you with that nice picture of some spandrels. This is from the Cathedral of Monreale in Sicily, and these are images of the story of Noah's Ark.
Thanks. I mean, there's a lot to talk about there. One of the I think challenges in the aging field is that people approach the idea of aging in a different way. I mean, I think that evolutionary theory has always been way ahead of the molecular theory, because I think originally when I got in the field, people were thinking of unicausal mechanisms of aging, DNA damage, or whatever your favorite model was, reactive oxygen species. And, as more and more evidence built up, there's a lot of different pathways you can look at, but there's, modifying an individual pathway only has a very small effect. And so I guess the question I would ask is, if this concept is right, what does that mean about thinking of things at a molecular level? Is it just a collection of bad things that happen as a result of antagonistic pleiotropy that drives aging? And if it is that, then, or hyperfunction, if it is that, is there any way to unify that in some concept that we can understand at the molecular level, or is it just a bunch of things that happen?
Right. I mean, the work that we've been doing has been looking a lot at individual diseases of aging, based on the idea that that there may be general principles that you can, that you can understand, that you can apply to understanding all of diseases of aging. It's not so much a theory that provides ideas about kind of changing aging as a whole. And that was what was so lovely about the damage maintenance paradigm, the old paradigm, when I came into the field, and the idea of disposable soma and so on. It looked like, according to those theories, that you could really go up to some kind of upstream cause of everything, like within cells, improve maintenance levels, and you'd basically eradicate, greatly delay everything, every aspect of aging. But from my perspective, that's really crumbled away thanks to excellent work, during the noughties and 2010s. It's like the field kind of tore out its foundations from underneath it, right? So I think that my sense is that overall the prospects, certainly looking at it from the perspective of the programmatic theory and related theories, the prospect for being able to intervene in aging as a whole look not good at all. And I suspect for humans, it seems that a lot of the plasticity in aging that has fascinated us so much, like especially in C. elegans, a lot of that is stuff which is not really, that you can't take that through to humans. There are odd things going on in these short-lived organisms, things involving torpor and seminal parity and things like this. So what it does mean, I think, what's great about these emerging theories is the possibility of actually finally understanding the origins of diseases of aging, actually having general models of aging-related disease to be able to figure out how to intervene in them. But in terms of the old idea, from things like calorie restriction and long-lived mutants and so on, I think the prospects have kind of look less and less likely of being able to translate that to the things that we knew years ago.
I want to get to that, but before I do, this maybe stay on the other topic, which is, this theory explains age-related disease as well. Does it explain like the normal functional changes that happen with age? Like you get older, you're cognitively less functional, you don't run as fast, your resilience is down. Is there any difference there between that and like pathology that is clearly a deviation from normal function?
Yeah, I think what the programmatic theory would argue is that there isn't really a major difference, except that some of them are more universal than others, right? That ultimately it's all the same thing. It's all senescent pathology of one kind or another. So there are some senescent pathologies that run away and become lethal ahead of all the others. But ultimately, you're looking at degenerative processes, a family of degenerative processes.
So, when I look at the antagonistic pleiotropy and this theory, which I think is very elegant, when I look at it from a perspective of intervention, I have a more positive view of it, because you mentioned the fact that a lot of this antagonistic pleiotropy is conditional. So it can be temporal, it can be sex specific, it can be environmentally specific. And so if you take the temporal example, you need a lot of TOR signaling when you're in development. You don't need a lot of TOR signaling all the time when you're an adult, and especially later, and yet it's happening. But that gives you an opportunity. I'm using TOR as an example. We could talk about many other examples, but you can turn down TOR later after development, and you're not paying the cost, at least at the primary cost, and you may be getting the benefit. What do you think about exploiting this antagonistic pleiotropy?
Right. I think the fundamental question really with this sort of what you could see as a kind of shift in paradigm away from damage maintenance, which it's not that damage maintenance has no role in aging. I think what the theory is arguing is that the other thing, the biggest part, if you like, is the programmatic part. Exactly. So in a way, there are two things there. One is that you have context specific effects of genes where you have beneficial in one context, harmful in another. So the sort of original context specificity was good early, harmful later. So there you have a possibility of dealing with, especially with hyperfunction, you've got the possibility of knocking it down. So knocking things down is sort of pharmacologically not so difficult. With hypofunction, a little more difficult. And the other side of it is that understanding antagonistic pleiotropy is also very much about it's about understanding mismatch between evolved gene function and the conditions of life, the way we live. So I mean, we have things like the massively obesogenic diet across the world, which we're not evolved for, and so our wild type sort of glucose homeostasis functions don't work. You get diabetes because it's a mismatch. Or, for example, there's the whole hygiene theory. The idea that a lot of inflammatory diseases are growing in frequency because we've lost too much. There's too little exposure to some of the kind of pathogens and so on that we've evolved our immune system has evolved in a situation where you have a lot of pathogens around, right? But I mean, I think those things don't necessarily they're not so easy to lead to interventions, I think, right? In a way, that's not the obesogenic situation is more of a social issue, a political issue, I think, than a
but mismatches in theory, well, first of all, mismatch is going to dramatically change the constraints that are applied to the organism. And of course, that may not make much difference over evolutionary time. But in terms of interventional space, if you could understand how constraints change, that may create opportunities. We don't live in the wild anymore. I say this all the time. People argue that we shouldn't study animals in a laboratory setting because they're not in the wild. And I'm like, I don't know if anybody's looked, but we're not in the wild either. I think we're probably closer to lab animals these days. So the constraints are different. And that means that the gene actions are going to be different as well. Question is, how do you understand that?
I mean, to my mind, what the field. I mean, I'm basically an experimental biologist primarily, and I test theories and using the worm, which are very versatile. And the damage maintenance paradigm was like a sort of a non-starter paradigm, and it was guiding research, and the research was going nowhere.
Yeah.
And the role of the paradigm is to essentially generate hypotheses that people can use to design their experiments, and that's going to lead them somewhere good. And I really think that, in a way, to my mind, key to understanding a lot of diseases of aging, and I think it's one of the most useful things that biogerontology can do is to provide understanding of diseases of aging, because aging causes these diseases. And at the root, I think, according to programmatic theory, this new constraint idea, is that to understand a lot of these diseases fully, key is understanding the nature of the constraint that's given rise to them, right? And once you understand that, then at least you can think about possibilities for intervention. If you want to treat a disease, you have to understand it first, or at least.
That's a reductionist approach, and I think it has a lot of value in that context. Is it possible to do a more synthesis-based approach to try to model what constraint looks like at an organism level? I don't know if it's tried to do that.
My own impression, and this is something that's kind of expressed in the book, is that when you start looking in detail at real examples of antagonistic pleiotropy in human disease, and you look at the way that the different constraints are all interconnected, it's like a nightmare. It's a sort of like a hell underworld of things all tied together, desperate kind of compromises that are made. It's like evolution is so hideously constrained. It's trying all the time to fix things, and every time it's screwing something else up. So, I mean, I wonder. There's been a history of sort of what do you call it? Sort of modeling aging, which I think hasn't been so successful in the past. But perhaps with a lot more knowledge, then yes, with a lot more understanding.
I wonder if this is a. Go ahead. I'm sorry.
I'm just saying the theories are very new. I think they're so interesting. I'm not even. A little part of me is not even sure. I don't quite believe them because they're too new, and you know what I mean. I say just you put them out there and let people play with them.
At some risk, I'm going to raise the following idea. Maybe these black box approaches that try to integrate complexity using AI might be better at trying to model a holistic view of what constraint looks like in an organism than using a reductionist approach. I don't. This thought just occurred to me. I'm not really even advocating it. I'm trying to wrap my head around the big picture. And, I understand addressing the specific issues that cause disease, but I guess I can't get away from the idea of some sort of broad general understandings.
No, I think I really disagree with you there. I think this is a broad understanding, because I think what we have here is the possibility of being able to look right across the animal kingdom at all of the diseases of aging that happen, and to essentially reduce them in a way that's a little bit analogous to the periodic table, where you have a certain number of elements that you can identify as working together in a complex fashion, right? And you can therefore you can establish fundamental principles that allow you to understand the whole spectrum of diseases of aging, where they come from, and how it links to the evolutionary process, because the evolutionary process is the main cause of disease. It's aging is primarily kind of an evolutionary disease because of the sort of bungling nature of the evolutionary process, if you like. So I think there is a global understanding here.
Yeah, it's a theory that could explain the diseases, yes. And I think the question I'm trying to get into, and maybe not articulating it well, is that what aging is? It's just a collection of these things happen that drive different diseases, or is there something more integrated to it?
Okay, so if you ask me, I would say that what the programmatic theory, as I see it, is saying is that potentially you could understand it purely as a whole bunch of genetic diseases, effectively. The question that I think remains open is whether there is some kind of overall timing mechanism, rate of living, if you like, not the Pearl rate of living theory, but something general, like a developmental rate of living effect that is controlling the rate of the overall thing. That, to me, that's a really interesting possibility, but I can't see it at the moment, right? I think that's an open question, and that, of course, would take us back to the old biogerontology dream of being able to intervene in overall aging rate. But
maybe that's something like entropy. I mean, maybe it's some physical process that's
very much. I think it's much more likely to be something that's just programmatic. There's something that controls the rate of development and the rate of progress of events in general.
Yeah.
I don't think you need to go back to the entropy idea.
Anyway, so I could do this all night, but we got a bunch of questions. So I'm going to bring American, who's going to collate them and try to find the questions that are hardest to answer. That's what I told him to do.
Good.
Thank you, Brian. So let's start right away. There are a couple of questions. Let me start with this one. Do WT genes contribute to the aging process when silenced?
I think I kind of answered that with what I was saying, talking to Brian just now. So, I mean, in principle, some wild type genes, if they are sort of classic hyperfunctional genes where they just continue on in a harmful fashion, then silencing them is a way to intervene in aging. So one could argue that, for example, rapamycin might be working in that way by suppressing various quasi-programmed pathogenic processes in later life. So yes, I think so. I mean, maybe not silencing them altogether, but probably lowering their activity level to one that's not pathogenic. I think if you knocked out TOR, for example, altogether, that would be quite harmful.
There's a couple of AI questions. So let's start with this one. Could AI help to predict spandrels in genes in animals and humans that were not observed yet in the young age or at the young age?
Yeah, I mean, my feeling about, this whole question of what used to be called systems biology of aging, it was a fashion in Britain for sure in about 10, 15 years ago. And my problem with it all was that you need to have enough input information to be able to do that kind of modeling. And certainly, I think the systems biology of aging, from my point of view, was a complete failure. But AI, of course, is unpredictable. I mean, you never know what AI is going to be able to do. I would hope that if the spandrels idea is correct, I mean, we've essentially been road testing it and it looks really good. But if it continues to be good and one gain more and more information, then in principle, yes. But I think you'd have to discover a lot about spandrels first and then put that information into AI. I don't think I'm never sure quite what AI is going to be capable of doing. But I think you need a lot of information to put into AI before you can get it to do smart things like figuring out tertiary protein structures from primary structure. Right? There's a lot of information.
I think you highlighted the challenge with systems biology, and I had a grant to work on systems biology. We published some cool papers, but I don't know if we did systems biology. But I think the challenge is the variables are just, uncontrollable. There are way too many variables in a biologic system to try to explain things. And so unless you're talking about a very, very simple interaction, it's almost impossible to apply a systems approach to it.
I totally agree. And I mean, I'm always sort of ready to be proven wrong, but that's maybe because of the whole systems biology thing over here, which I think was a bit of a fiasco, really.
Okay, let's proceed with the next one. How do you see if age-related diseases are the consequences of aging, and if healthy aging populations without any diseases can be the best subjects to study aging itself?
Right. So I think that there's a question about the idea of healthy aging without disease, which is a complex one. So normally speaking, people who age well, what actually happens is that they remain very independent and with a good quality of life up to a late age, but usually they become very ill at the end of their life. And occasionally people will die suddenly from strokes. If old people are found dead in bed, there's no autopsy done on them. So I think on the whole, the idea of people dying of healthy aging or sort of dying, so that people sometimes talk about people dying of pure aging without disease, is to a large extent something which is not real. On the other hand, there is this old analogy of the one horse shay, where you have, it's an old kind of conceit that used to describe dying from frailty, really, where you have an elderly individual that has many, many subtle things wrong with them, but none of them are actually evident in terms of actual pathology. And then you have an extremely rapid collapse, like everything collapses all at once. So sort of death from sheer frailty. But even there, I mean, normally what happens there is somebody will, with a good health in the 90s or whatever, they'll have a fall, and then they go into hospital, they get an infection, and they die within a week or two. And in a way, it's because there's just a complete collapse. So in all these cases, the death is the result of massive pathology. It's just that in some cases, it's not. It's that each individual system has deteriorated in such a way that you don't see clear symptoms until quite shortly before death. So I don't really believe in this idea of death from pure aging. I think it's a convenience from the medical world, which is, in a way, it's a way for clinicians to be able to deal with elderly patients. It's a very humane way to think about aging as sort of, natural death and so on. The Queen is supposed to have died of old age, without any disease. It's wouldn't have been decent to do an autopsy and find out that she died from a stroke or something. Why do that?
Let me move to something different. Taking the idea of spandrels and aging as a side effect and zooming down to the cellular level to a specific question, is the idea of spandrels then the stem cells, induce or otherwise, have somehow escaped these effects?
Right. So I think it's difficult because that's a very specific question. The question is whether stem cell failure, or inadequacy in later life, is a consequence of antagonistic pleiotropy, and I just don't know about that. I mean, one would argue if it is, it could in principle be something that's a consequence of antagonistic pleiotropy, where you've got something that's regulating stem cell renewal, which is subject to antagonistic pleiotropy because of constraint. But it might be something. I don't think what the constraint theory isn't saying is that all of aging is due to constraint. It's saying that it's a major element of what's giving rise to pathogenic effects of wild-type genes. There might be other things going on with stem cells. I don't know. Maybe it's a hypofunction issue. Maybe it's an impossibility constraint. The theory is not saying that the constraints antagonistic pleiotropy is everything. It's saying that it's proposing that it's a big element, but there's huge numbers of examples of antagonistic pleiotropy in disease, but many aspects of disease it's not clear how constraint or antagonistic pleiotropy play a role. So this is the beauty of a good paradigm. It sort of says, why not look at that? Lots of good work to do, right? Lots of grants.
I think that would lead to the next question. So, or your opinion actually. So the spandrel theory depends on the harmful effects being inseparable from the beneficial ones, but this is not necessarily the case given enough evolutionary pressure. So what's your opinion on that?
Right. So, I mean, in a way, what one has to reconcile with the spandrel theory is the fact that the aging rate evolves very, very quickly, right? So, for example, if you just think about humans and our sister species, chimpanzee, I mean, our longevity is something like almost twice that of the chimpanzee, and the common ancestor with the chimp was around in evolutionary terms very recently, really about 5 million years ago. That almost suggests that somehow the whole aging process has just evolved at a different rate somehow. And this brings me back to what I was discussing with Brian, which is that I do wonder if there aren't still some fundamental underlying determinants of overall aging rate, which determine like when these antagonistic pleiotropy effects appear. I mean, as an example, we've been looking at trying to use these programmatic theory and the wider theory called the multifactorial model to try to make sense of diseases of aging. One of the ones I looked at was osteoarthritis. What's so striking about osteoarthritis is that so many mammals get it, and yet they get it at the same sort of roughly the same proportion of their adulthood, right? So they'll all get it in later life. Clearly there's something similar going on in each case. So if one argued that it's a consequence of antagonistic pleiotropy, the timing, or the relative timing, hasn't changed. It's just that the overall lifespan has changed. So I think I really wonder about that. I think that it seems an unknown, right? Because if it was things were so constrained, how would you get such rapid evolution of the whole aging process, right?
One more quick question. This is slightly different to the other ones. So what do you think is the best way to build someone's own understanding or pathway of aging research?
It's terrible. It's terrible for anybody coming into the aging field. Conceptually, the aging field is more chaotic than it was when I came into it in the 90s. There was a sort of consensus in the 90s about a lot of things. So now it's awful. And I don't know, I think I could say, I mean, my book is an attempt at a portrait of the whole field. And it gives a kind of global sense of where the field is and how things fit together, but it's only one person's view. I think the most of the books on aging that there are tend to be relatively narrowly focused, or they just reflect the disjointed nature of the aging field. It's like a series of fragments, like islands of knowledge in a sea of the unknown, not connected together. So it's difficult, very difficult. But yes, maybe my book will help, or maybe not.
Let's hope that.
Get it published as quickly as possible. We'll have you back on to talk about the book maybe when it comes out next year. So
yeah, happy to do that.
Thanks a lot, David. And thanks, Merrick. I
Thank
want to
you.
remind people to use the chat function and the panelists and all attendees option to leave comments. We have a big seminar on supplements coming up February 29th and March 1st, Unlock Healthy Longevity. Please look for the QR code and sign up for that. I also want to point out that you may just see me and a few of the questionnaire people on the show, but there are actually thousands of people behind the scene that make this show great. One of them, who's one of the key members, is So Young Sun. This is his last show, and so I want to thank him for everything he's contributed to putting all these shows together. We don't really have thousands of people, but it is a lot of work, and so he's done a great job. The next episode will be November 16th, and our speaker will be Charlie Brenner. That should be interesting if you know Charlie. And I'll be looking forward to a discussion with him. His topic will be on NAD coenzymes are disturbed in diseases and conditions of metabolic stress. So we'll be talking about NAD and sirtuins and a lot of other things, I'm sure. So I'll be with you next week. I'm going to leave you with a final video about an 80-year-old who still feels spandrel free. See you next week.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.